Hey, good morning, everyone. Happy Friday to you. So today we're going to talk about ATP a bit more and the role of ATP in our cell's metabolism. But first, I wanted to finish up our discussion about enzymes that we started on Wednesday, and then we ran out of time. And so why don't we first wrap that up? And it all ties in together because much of our discussion about enzymes and how they work leads into our discussion about the importance of ATP as an energy molecule that, among other things, powers those enzymes and allows them to work. Okay. We are talking about enzymes as being catalysts, meaning enzymes, these molecules that most of the time are proteins, right? So most enzymes are actually proteins, and that makes sense because proteins are a wonderful type of macromolecule that gives you lots of flexibility over specific well-defined shapes, right, of something like an enzyme. We said that enzymes work as catalysts by lowering the activation energy needed to make some other chemical reaction happen. Recall that we said that all of the molecules that are used in our bodies are what we call metastable, meaning that all the molecules that our cells use are stable enough that they're not just going to spontaneously fall apart and have all their bonds break, because then you'd have no control over anything, and that wouldn't be compatible with life. At the same time, they can't be too stable, because the more stable a molecule is with all of its bonds, the harder it would be to actually undo or break those bonds, right? So the molecules that we have in our cells are sort of like that happy in-between of being metastable. They're stable enough to potentially last indefinitely, but yet it doesn't take too much to get those bonds to break when your cell needs to break them, right, for different biochemical reactions. We said, just like the example of the boulder or the ball on the top of the hill, an existing chemical bond can be thought of as potential energy, right? The electrons in that bond, they're stable in that bond, just like the ball could sit on the top of the hill forever, like 10,000 years, right? But at the same time, that potential energy can be turned into kinetic energy. In the case of the ball, we're talking about gravity being the sort of motivating force that pulls it down the hill. So if you put in a little energy to get the ball rolling, the ball rolls until it reaches the bottom, right? And all of the potential energy was converted into kinetic energy. Now with molecular bonds, it's not gravity, right? This whole ball on the top of the hill thing is just kind of like an analogy to get you to think about energy. But in chemical bonds, the potential energy is the sort of energy of the electrons in there and what state they're in, right? And the fact that if those electrons can be encouraged to momentarily break free of that bond, if there are other atoms around that the electrons could form better bonds with, that put them at a lower energy state, right? That's the kinetic energy being released, okay? And then the better bond at the end would be like the ball arriving at the bottom of the hill. Meanwhile, that difference in energy of the bond being broken compared to the one that's now formed is released as energy that can be harvested or lost as heat, right, into the environment. So in the example where we looked at ATP, we said that if ATP starts off at this level of free energy, right? G, capital G, means how much energy is in this bond of the third phosphate being attached to this ATP to make it a triphosphate, right? Compared to the bottom of the hill is when you've broken off one of those phosphates, the third one, and you're left with ADP, right? Adenosine diphosphate. That molecule, the ADP, would have a lower energy associated with it, right? Importantly, we said, just like the ball that's stable at the top of the hill, this metastable ATP molecule could stay as ATP forever, right? It'll happily float around as ATP, right? So you have to actually put in some energy in order to agitate the electrons to get the bonds to break. And that is the energy of activation. Right? So that's the guy having to push the ball to put some energy in to get it going. We said that enzymes function by lowering how much energy you have to put in, making it easier to break the bond. Right? So substrate activation, remember, substrate means whatever the enzyme molecule is working on as its target. So for example, an ATPase enzyme would work on ATP as its substrate. The activation of the substrate can happen in three common ways. One way that we discussed was that the enzyme holding on to the target substrate might actually torque or bend the bonds in the substrate, thereby making it easier for the bonds to break. So having these bonds torqued in such a way means less energy would have to actually come into it now to actually make the bond break. In some cases, enzymes do undergo chemical reactions with the substrate, for example, donating protons, or maybe the transfer of electrons, making some sort of temporary covalent bond with the substrate. So that leads us to this slide. This equation is a pretty general equation. It's not meant to be kind of mind-boggling difficult or anything, but it suggests that when an enzyme, E, and its substrate come together, S, right, there's going to be an intermediate sort of temporary form where this molecule is now the enzyme and the substrate interacting, right? Either the substrate is just fitting into some sort of activation pocket on the enzyme, or like we said in the previous slide, there might actually be a covalent bond temporarily formed, right? But that puts the substrate in this activated state, right? That's more likely now to require only a little bit of additional energy to get something like the bond to break. So then it would become the products of this reaction, P, and importantly, the enzyme is not used up, right? That protein still exists, even if it's gone ahead and taken a small substrate and broken it up into two products. Right? Here's an illustration to help you visualize that. This whole blue shape here, right, that looks like two circles together, this, let's pretend, is one big protein, right? So if you were to unravel this, you'd find out it's just one long polypeptide chain, but now it's this big globular three-dimensional shape. And for ease of simplicity, the cartoon just shows it like this. Right? That surface of the protein would have many different types of three-dimensional features on it. Let's say one particular feature would be the active site, where the substrate that this enzyme works on is going to bind. Right? Notice, though, in this particular shape, the substrate, the active binding site for the substrate, looks closed, right? Therefore, the substrate won't fit in there right now. And this enzyme might be inactive, right? It's not working. Somewhere else on the enzyme, it could be very close to the activation site, it could be actually part of the activation site, or it could be somewhere far away elsewhere on the enzyme protein, is another three-dimensional feature. We call this the allosteric site. What does allosteric mean? We've brought this up before. When a change in the shape of one region of a protein impacts a change in the shape of another region, right? So because the protein is just one long polypeptide chain that's wrapped up, and all of the amino acid side chains of those residues are interacting to kind of find a happy shape where they can all kind of coexist, right? Because they're bumping into each other, repelling each other. Some of them are attracting each other. If you change the shape of one part of the protein, there might be a ripple effect, right? That causes the rest of the protein to have to rearrange and change its shape as well. So enzymes take advantage of that. For example, we have this inactive enzyme at the start here in the cartoon, but let's say some other molecule comes and binds to that allosteric site over here. Right? Here, we're calling it an R, for this is actually sort of like the regulatory region of the protein that causes the protein to change its shape. And look what happens to the active site. Its shape changes also and opens up. And now it's the right shape for the substrate, S, to now bind there. Okay, and now the enzyme will go ahead, activate that substrate, because now instead of just the E plus the S separately, it's now this intermediate ES, right? And then it catalyzes the reaction and you get the two products. You know, maybe, for example, the substrate gets split in half, right? That's why you now have the triangle part and the round part. Quite often, this green allosteric activator is something like a phosphate group, right? Because phosphate a phosphorus with 4 oxygens around it. It's a pretty bulky little molecule. And we've already learned that oftentimes phosphate groups can be added to proteins in a post-translational modification. So a phosphate group gets added to a protein, and that might actually be the thing that then activates the protein or the enzyme. Or it may be that normally the enzyme shape is active to start with, but your cell needs to turn it off. So maybe by phosphorylating it, it changes its shape and it turns the protein off. Okay, so phosphorylation is a very common, very important way of controlling proteins that are things like enzymes. This is a slide we looked at last time, but I think this is a good opportunity to revisit it. We said that enzymes are very specific for the particular substrate that they evolved to work on. We said, let's imagine at the top of the slide here that you have this particular small molecule that needs to be worked on, right? In this particular case, let's say your cell has some use for this molecule, but the problem is this molecule starts off with this blue oxygen atom attached to it, right? And as a result, the shape of this molecule isn't the right shape, or it doesn't have the right properties for whatever intended purpose your cell has for it. So in order for the cell to make use of it, you need to take the oxygen off. Again, this little orange molecule with the blue oxygen on it is metastable, right? Meaning that it could exist like that, indefinitely. But at the same time, there's nothing that says the oxygen has to stay on it, right? The oxygen can be taken off, but that just won't happen by itself, right? Potentially for like 1 million years, right? So if your cell needs to make use of this process to get the oxygen off, this is where enzymes come into play to really speed things up. We said that here, this big blob, right, is this protein that's functioning as an enzyme. Again, imagine that it's just a big, long polypeptide chain that's all rolled up into this globular shape. And this particular shape, right, is due to the specific amino acid sequence of this protein. Right? Importantly, here is the activation site. So there's a pocket in here. And if you were to look at the amino acid side chains of the amino acid residues of the protein that are kind of sticking into this pocket here, that's what we see on the left. Right? So these purple side chains are the amino acid side chains that are positioned to actually stick into this pocket, thereby defining the very specific shape of that pocket in there where something can bind. And in this particular case, that substrate molecule, the one with the oxygen on it, binds perfectly well in there, right? Because look how nicely the amino acid side chains that line the pocket define, right, the possibility of this substrate fitting in there and binding, right? It's just like a key opening a lock, right? How perfectly matched that key has to be to the space inside of the lock in order for it to work, right? So when the substrate binds in there, then there's the opportunity for the enzyme to go ahead and twist the shape of the substrate, maybe putting a lot of torque or strain on the bond between the oxygen and the other orange molecules in there, thereby making it way more likely that the oxygen can then break off, right? Okay, so instead of taking 10,000 years for the oxygen to come off, if left to do it by itself, maybe now it comes off in literally like a millisecond, right? Once this enzyme gets a grip on the substrate. See how the enzyme is moving? The enzyme is changing its shape in order to cause the pocket to change its shape and bend or twist the substrate. Right? So this is what we're talking about enzyme control. Imagine, if you will, up in the upper left part of this enzyme, you see this pocket that's opening up over here, right? Like this particular side chain of an amino acid gets pushed off to the left, opening it up. It could be that, for example, this is due to a phosphate group that is being added to this particular amino acid side chain, because then you have a bulky phosphate group on here. It causes the side chains to have to shift. And that shifting at this regulatory site, right, causes a ripple effect through the whole protein, right, that then causes eventually, right, the binding pocket to twist like this, thereby catalyzing the reaction. Okay. So that's why the addition of something like a phosphate group to this protein right at this spot right here, where I have the cursor, could be the way that this enzyme is controlled. Right? We would say it's controlled via phosphorylation. Right? Okay, so here's just a quick thought question before we move on. Why are enzymatic proteins then, like this one in this example, very sensitive to temperature and pH? Well, we learned, remember when we first started learning about DNA replication, and we talked about DNA polymerases in our cells, right? How they are optimized to work at like pH 7 and also at 37 degrees Celsius, right? Because that's the temperature of our body cells inside of our living body, right? We said something like an extremophile bacteria, like T. aquaticus bacteria that lives on hydrothermal vents at the bottom of the ocean, where the water is scalding, right? And the pH is kind of different, right? It also has DNA polymerases that also copy its DNA. And yet, right, those DNase polymerases work at like 90 degrees Celsius, right? And what's the difference? The difference is slight variations in the amino acid sequence of the Taq version of polymerase versus the human DNA polymerase, right? They're close enough that you would still have, for example, the equivalent of a binding pocket on them that's well formed to specifically bind to things like DNA and nucleotides to be able to add nucleotides to the DNA, right? But maybe the other amino acids vary so that the shape of the protein actually still holds together well at 90 degrees in the extremophile bacteria, right? But in our bodies, our version of the Taq, right, holds together best at 37 degrees. Okay. All right. So let's continue talking about phosphorylation. So we said that phosphate groups are often transferred, for example, taken off of an ATP and added to a protein in order to ultimately drive an enzyme to change its shape and do its job. We said that, for example, the various types of active transporter proteins that are in the membranes of our cells that we learned about before the last midterm, and how they have to use energy to push things against a gradient across a membrane, those, we said, are oftentimes ATPases. So there you go, right? Those are examples of enzymes also, because they do something or they make something happen that, if left alone, wouldn't happen by itself, right? And we said that many of those transport proteins, they bind ATP, break ATP, right, to make use of the energy released by ATP to change their shape in such a way to maybe push some substrate or a solute against its gradient. Okay. So proteins, enzymes that actually take the phosphate from something and add it to another target, right? For example, one protein adding a phosphate group to another protein in order to turn on that second protein. That first protein that does the transferring of the phosphate group are called kinases, right? The kine in kinase, I think, is the same as kinetic, right? Because stuff is happening as a result of these enzymes transferring phosphate groups to other targets. They are phosphorylating other targets. At the same time, right, you might have a target that has a phosphate group on it, like an enzyme that has been activated, right? And now it's doing its thing, but maybe you need to turn this enzyme off again. How would you do that? Well, maybe you remove the phosphate. How would you do that? It would involve another protein that specializes in binding and removing that phosphate, right? And that second protein would be called a phosphatase. So look at this example here. Kinases are a highly evolutionary conserved family of proteins, right, that bind ATP and potentially some other substrate at the same time. They break the third phosphate group off the ATP and transfer it onto that other substrate that they're hanging onto. And now that other substrate has been phosphorylated. Right? If we look at an example of what a kinase's activation domain looks like, this is what we see here. This is not the entire protein, right? This would not be the entire, for example, big purple blob that we saw animated in the previous slide. This would just be zoomed in at the activation site, right? And so you can see here are the surrounding polypeptide regions of the protein. It's not showing you the actual side chains of the amino acids because that would just kind of get in the way. But what they're showing you is in this particular case, here's a molecule of ATP, right? So here's ATP, right, with its 3 phosphates, and it's bound in here. It's kind of hard to make out, but the green dots are the carbons that make up the various rings here. And then the red and purple are the 3 phosphate groups sticking out here, right? So this would be just the right shape binding pocket to bind ATP and encourage that 3rd phosphate group to break off. At the same time, this pocket is shaped so that some other substrate can bind at the same time, maybe some kind of a target protein, right? So that when this kinase protein binds the ATP, breaks the phosphate off, it's also the right shape to transfer that phosphate onto this other tan-colored substrate, whatever this one is. So that substrate, if it's a protein, floats off, and now that protein is phosphorylated, right? So I want you to think about the specific shape here. What parts of this shape must be conserved in order through evolution to still have a functioning ATPase activity? And what parts of this shape are more variable to allow for a different assortment of different types of substrates that can be acted on by different types of kinase proteins, right? So as an example here, let's think about a stapler, right? Because after all, an enzyme is a great example of a protein functioning as a small molecular machine, simple little machine, right? Hey, so a stapler is like a very simple little machine, right? Hey, we're all familiar with staplers, right? Let's think about the construction of a stapler. How much of what you see in the overall shape of the stapler, right, is really set in stone? Meaning, if you were to build a new stapler, what parts do you have freedom to kind of play with in terms of the shape? What parts do you have no freedom to play with if you want it to be a useful stapler? Let's consider that the activation domain of the stapler is this part here, right? And what does it do? It takes staples, right? And the other substrate that might be binding at the same time is the paper. Okay. So I want you to consider this. When you go ahead and you have a stapler, right, and you want to staple some pieces of paper together, think about it this way. This is a good example of an enzymatic reaction. I have several pieces of loose paper here. These were just dropped off by a colleague of mine because I'm supposed to review the molecular cell biology part of our Manoa catalog. So these are loose pieces of paper. If I wanted these pieces of paper to stick together, I could, for example, stick them together, maybe put them outside, put a heavy rock on one corner, right? And then maybe 10 years from now, if I go back and pull this out, I will have found the fibers of the paper have then joined in the corner, right? Now, what if I don't have 10 years to wait around? What if my colleague is expecting me to finish proofreading this by the end of lunch, right? That's where a stapler comes in useful, because with a stapler, I could just go ahead and staple it. And now these pieces of paper that would have taken 10 years of having a rock sitting on top of them to get the paper fibers to stick together, I just took care of that in a split second, right? So the stapler is acting as a catalyst. Now you might say, what is the function of the stapler? Why was this stapler invented? You might say, oh, well, obviously it's to staple pieces of paper together, right? Because you would say, well, the paper is the only substrate, right? But that would be wrong, right? Because the stapler still works even though you don't have paper present. And that's when you realize that the real job of the stapler is not to staple pieces of paper together. The real job of this design is to bend staples, right? In other words, if I do this right now, right, and I get a bent staple, even though there was no paper involved in this, would you say, oh, the stapler is not working, it's broken? No, the stapler is working 100% the way it's supposed to be. In other words, it was invented to bend staples in a particular way. And the fact that you can actually have pieces of paper stuck in there when it's doing that, the paper is the secondary substrate. It is like a byproduct of the stapler's evolution. That's why, remember, one of the points that I was trying to make to you earlier was, why do we say ATPases are all just ATPases, even though they seem to do other diverse functions in the cells? Right? If you were to imagine the stapler as a protein that resulted from some genetic sequence, a set of instructions in DNA, right? What parts are you free to mess around with? Right? What parts have to be kept the same? The parts that have to be kept the same would be the parts that are specifically having to do with bending the staple. Right? So in the case of the kinases, it would be the sequence of amino acids that are involved in binding and breaking the ATP. Right? The other parts of the stapler are more variable, right? And that would be the more variable substrate binding site of what the secondary effect is. So over evolution, you could have many, many, many variations of staplers, right? Maybe starting originally billions of years ago from one gene, the original ancestral kinase gene that coded for a kinase protein that could bind ATP and break it, right? And then over evolution, because that's a useful gene to have, maybe through different types of mutations or mistakes in replication of these early cells, you ended up with many copies of this gene scattered throughout, for example, the DNA, right? And each of these encoded for this protein. But over eons of evolution, random mutations all through the DNA began to change those genes, right? And just as long as the actual kinase domain part stays the same, you could have variability in the rest of the protein. So think about it this way. Right? So enzyme catalytic domains are highly conserved because when evolution stumbles upon a useful protein structure, right, you see it again and again and again in the different proteins that sort of diversified from that. Different genes through evolution, right, that all started from that original gene, still keep the part that's most important, but then the other parts vary. So in other words, all of these staplers in their various shapes and sizes on this screen here, they all have the same staple activating site. That's why you can use a generic set of staples, right, to work in any of these staplers. But the rest of the stapler, the rest of the protein, is free to change. And if through evolution, that particular change that was introduced actually turns out to be useful for something else, then you have a different functional kinase protein. So here's some examples. These are three different kinase proteins. One is EGFR, which is an epithelial growth factor receptor. One is HER2. One is a CDK, or cyclin-dependent kinase. We'll go into these in a little bit more depth later on, so you don't have to worry about the actual specifics of them in this lecture. But this is the kinase domain. And you can see they're very similar in shape because they all bind ATP, right? That's how they evolved. That's the part that got held onto through evolution. But other sequences around them, right, the identities of the amino acids changed. And as a result, they can interact with different secondary substrates. So for example, the kinase function of EGFR is important for epithelial cell growth. Right? The function of HER2/neu, if you lose control of that, is implicated in the formation of cancers. CDKs are important. Their kinase activity leads to cell cycle regulation, right? And again, because the second substrate that they bind to, in addition to the ATP that they end up transferring the phosphate to, can be different molecules because of that variability in the kinase's shape, right? All right. So let's shift gears a little bit and talk about ATP, because after all, ATP, we've been focusing on it now as the source of somehow energy, right? Molecular energy, you break the phosphate bond and it's something that can change the shape of another molecule. Right? Think about metabolism. So life is one continuous chain of metabolic cellular reactions, right? Inside your cells right now, you have, an almost unimaginable number of different molecular pathways happening. Some of them are building up complex molecules from simpler ones, in which case they'd be anabolic pathways, right? And that would require putting energy into this. Other pathways that are happening simultaneously are catabolic pathways, breaking down molecules into smaller parts, for example, nutrients, right, into small raw materials. And those would be exergonic, meaning they're releasing energy as a process, in the process, right, a negative delta G. Let's think about glucose as a starting point here. All right, so glucose with all of its carbon-carbon bonds, we already said that if you oxidize glucose, meaning if you go ahead and remove electrons from glucose, that would be a negative delta G. You are releasing energy. Okay? And we said that if you took 1 mole of glucose and completely oxidized it down to CO2 and H2O, you would release 686 kcals per mole of energy from that. In our living cells, this would be the end of the line. Our living cells can't do anything else with this carbon dioxide or this water, right? So we get rid of the carbon dioxide and we also get rid of the extra water. Right? On the other hand, if you started with the glucose molecule, instead of oxidizing it, you actually had other enzymes taking the glucose and stringing them together. Right? So some other enzyme functioning as a carrier molecule that activates glucose molecules and actually polymerizes glucose, that would be a plus delta G reaction because energy would be required to do that. Perhaps each of those enzymes that are working on the glucose maybe have to break ATP in order to get them to change their shape and force glucose to be added on to a growing polypeptide chain. Right? So that would be the more reduced form of glucose towards being like the starch molecule. The more oxidized form would be heading towards the carbon dioxide and the water. Right? So glucose is the primary fuel source for our cells. I think you know that, right? Our cells are powered by glucose. But then you say, wait, I thought we were just saying ATP is like this fuel source. No, this is why we're going to very carefully make this distinction right now, right? Because I remember as a college student, I didn't get it. I would often hear that ATP is an energy molecule, but at the same time, I'd hear like glucose is an energy molecule. So what's up with that? Let's explain this, right? Glucose is the primary fuel for your cells, but the energy from glucose must first be converted into a more convenient molecular form that the various proteins in your body that are enzymes that require energy are designed to expect. Right? So think about this. Here's the burning of fossil fuels as an analogy. You have coal, right? Coal, of course, you realize is a fuel source. That coal goes to coal burning factories and is converted into electricity, right? And then the electricity is a much more convenient form of the energy that was contained in the potential energy of the coal, converted to electricity. It makes for much easier distribution all over the place, right? Ending up in, for example, an outlet in the wall of your house. And so the outlet, you can plug in something like your phone to charge up, right? So therefore, the energy in the coal was converted into a form that your phone is expecting to use as energy, right? If your phone is currently in the red, right, and it's about to die, and you tell your friend, oh man, my phone's about to die, and your friend said, don't worry, here you go, and she handed you a handful of coal, right? That's not going to help your phone. That is a lot of energy, right? But it's in the wrong form. It has to be converted into a form that can then be used by how your phone expects the energy to look. Right? And that's what ATP is, right? So the energy from glucose has to be converted into this energy molecule, ATP, that is easy to distribute throughout the cell. And importantly, as we've been talking about now for the last couple of lectures, many enzymes are designed to expect ATP as the thing that they bind to so they can break that third phosphate bond and cash in on that energy. So I think you guys already know this, right? But let's just quickly say again that ATP, right, is this nucleotide, adenosine, with 2 additional phosphates on it, right? Because if it was just an adenine with a ribose sugar and 1 phosphate group, that would be your classic nucleotide, right? But if you add a second and a third, now you have ATP. That third phosphate group, right? These phosphate groups are held together by these phosphoanhydride bonds, And these phosphate groups really hate each other, right? Because they're big, they're bulky, they're negatively charged. So they're repelling each other, right? They would actually love to be able to pop off from the third one popping off from the other two, but it's metastable, meaning the electrons that are involved, well, maybe they're not happy, but at least they're kind of like satisfied that at least they're in a bond, right? So the ATP as a result could last as ATP indefinitely. But that bond, that phosphoanhydride bond between the third and the second phosphates, right? That's a pretty high energy bond because of all those repulsive forces and the physical bulkiness of it. So then you'd end up with ADP and this free inorganic phosphate. And that reaction then of breaking the third phosphate group off of ATP releases, right, minus 7.3 kcals per mole of energy. Right? So that's the amount of energy then that something like a kinase enzyme that is bound to ATP and is breaking that third phosphate group off, that's how much kcals is being kind of cashed in by that enzyme, right? And that amount of energy then will then fuel that enzyme changing its shape. Right? All right, so if we say that glucose then is the raw material, the raw fuel for your cells, we said that glucose has to be converted into ATP, right? And we will learn about that conversion in the next few lectures. But once you have ATP, then all of the energy, that potential energy, right, of the glucose molecule, right, is now stored as small potential energy in each ATP molecule. And therefore, this ATP now goes all throughout your cell, and you have, thousands of enzymes, right, that all are waiting for the ATP to bind it, right, and actually do some sort of a function, right? All the type of cellular work that we talked about is powered by ATP, right? And so you can see that in this case, all of these different enzymes that might break the ATP, right, they all have evolutionarily conserved ATP binding sites, right? But the other parts of the protein might be very different so that they can then do something else with that energy. So that's why we say ATP is like an intermediate currency model or a molecule. Think about the value or the worth of one molecule of ATP in terms of energy. This table here, I know this looks a little intimidating. You don't have to memorize this table, but you have to understand what it's trying to illustrate, right? Here is a number of biologically relevant and important compounds that our cells use as energy, right? And each one of these has a particular given cash value, right? So ATP, for the sake of this illustration, they've put it right in the middle, because ATP, if you break the third phosphate group off, right? So you end up starting from ATP, you end up with adenosine diphosphate, ADP, and then a free floating phosphate that's broken off. We just said that releases minus 7.3 kcals of energy, right? That can be used to power something else. Other bonds breaking, for example, a glucose molecule, right, that has a phosphate group added to it on its first position. So glucose 1-phosphate, right? If you break the phosphate group of that off, that releases minus 5 kcal per moles. Right? Interestingly, if you had started from a glucose molecule that had a phosphate group on its carbon 6 position, right, and you broke that phosphate off, right, that releases less energy, minus 3.3. Why? Because again, the position of the phosphate group, right, and how unhappy it is being stuck where it is, translates into how much energy you can actually get out of it. Right? The other direction, other bonds, for example, this 1,3-bisphosphoglycerate. Right? If you break the phosphate group off of that, so you end up with 3-phosphoglycerate instead of a 1,3-bisphosphoglycerate, that liberates minus 11.8 kcals of energy. So what does that imply? Remember, if we say that an ATP breaking the third phosphate group off releases minus 7.3 kcals of energy, how much energy would it take to take a free phosphate group and actually force it back onto an ADP to make an ATP? I think many of you know the answer. It would be the same amount, except for it would be then plus 7.3 that you have to invest in it. where would that energy come from? It would come from some other, right, bond or something like that, that is providing more energy than the 7.3, so that you can actually take that energy and invest 7.3 kcals worth in taking a free phosphate group and shoving it onto an ADP to make an ATP. Right? That also means what? If you break one ATP and release 7.3 kcals of energy to play with, that's not enough energy to do something like build a 1,3-bisphosphoglycerate, right? Starting from a 3-phosphoglycerate and a free phosphate group, right? In other words, you wouldn't have enough energy released by the ATP breaking here so that this phosphate group coming off of it could be this phosphate group here being shoved onto this to make the 1.3, because that bond energy is higher. It's more expensive, right? That's why these molecules are arranged that way, because what it's basically saying is the breaking of one of these more expensive bonds provides enough energy to form any of the less expensive bonds. Likewise, what this also tells you is that the cashing in of one ATP molecule, right, would be enough for you to form any of the less expensive molecules underneath it, but it's not enough to form any of the more expensive ones. It's like the currency. So $1, right? The exchange rate with the British pound, I think it's sort of like, I don't know what it is, $1.50 to get you 1 British pound. So if you wanted to convert the currency, you could not convert $1 and get 1 British pound. You would need at least $2 to get your first British pound, right? But certain currencies, like this yen over here, right, they're cheaper, they're lower valued currencies. So $1 could be cashed in to get you many yen, right? Okay. So let's think about this flow of energy, right? We always said that energy flows from uphill to downhill. And part of this is understanding how heavily reduced molecules in biological reactions can be oxidized in different steps to get more oxidized molecules, right? At the end, right? So let's look at an example. Here we have ethanol, right? This is the kind of alcohol that you drink. So if you oxidize alcohol, you can convert it into acetaldehyde, right? So in this case, by having this oxygen engage in a double bond, the oxygen is pulling the electrons away from this carbon that's in the middle of the illustration here. So that carbon is becoming more oxidized, right? In the process, you would also release a couple of hydrogens over here, right? So the removal of electrons, it's never just one electron that's taken as part of oxidizing something and reducing something else. It's always a pair of electrons, because electrons travel in pairs. Not only that, but you're not going to have two naked electrons with their negative charges being sort of moved around. They will always carry with them 2 protons. So usually a pair of H's, right? Or H pluses. And so as a result, that's why you end up with, right, a molecule of hydrogen, right? 2 hydrogens, because they represent the 2 electrons that came off of the ethanol, right, with the 2 protons that travel with those electrons. So remember, this hydrogen pair right here, it's not actually released. It is being transferred to another participating molecule, right? That molecule is oxidizing, for example, the ethanol and turning the ethanol into acetaldehyde, and that molecule, as a result, is receiving that pair of electrons and pair of protons and is becoming reduced, right? So that's a very important thing to keep in mind, right? So consider this. If enzymes often work by doing that, right, by things like taking electrons off of a molecule, thereby oxidizing that molecule, converting the ethanol into acetaldehyde, if that enzyme that does that is now stuck with a pair of electrons on it, right, it can't work on another molecule of ethanol until those electrons are taken off of it, right? So it can go and grab another pair, right? So what would that involve? Well, you could say, well, that would involve another enzyme that takes that pair of electrons off of the first enzyme and transfers it to the second, right? And so on and so forth. You can already tell that would not be a very efficient way of dealing with this, right? Because you'd have to always have the next enzyme that takes the electrons off of the previous enzyme, right? So what's a better solution? The better solution is enzymes often use another small molecule as the electron acceptor. In other words, as that enzyme is taking electrons off of a substrate, those electrons don't end up on the enzyme itself. They get instantaneously passed or dumped off to some little molecule, right? Which is not a big enzyme, it's some little molecule that can just float away and be used as something else. So we call these electron acceptors. These electron acceptors, because they help enzymes work, right? Oftentimes enzymes won't work unless they have this other small molecule to dump electrons onto. We call these coenzymes, right? And they serve as electron acceptors. So coenzymes are small molecules that bind to an enzyme at the same time as a substrate, right? And they serve as where the electrons that are taken off of the substrate can be dumped so that the enzyme then lets go of both things and that enzyme is free to work on another substrate, right? It's not burdened by the two electrons that it took off, right? Here's an example. Nicotinamide Adenine Dinucleotide, or NAD+. This is the structure on the left, right? It looks kind of weird, but you suddenly realize, well, there's something familiar about it. It's two nucleotides together, right? So here's an adenine, a ribose, and a phosphate group, right? So this is an adenosine nucleotide, but it's linked between the two phosphate groups here, right? Directly between the two phosphate groups to this other nucleotide. And this nucleotide is a little funny. Its nitrogenous base is this nicotinamide group. Here you go, another example of another nitrogenous base that's not an A, a T, or C, or G, or a U, right? And in fact, this is a very important one. So nicotinamide is called this because if you look here, this is nicotine, right? And this is the structure of nicotine. So if you take this structure and replace this group over here with this group over here, right, you get a nicotinamide group. And so this group over here, very happily, so it's called NAD+, it very happily will take a pair of electrons with the two protons that are traveling with those electrons and become NADH, right? So you can see those electrons got added over here along with one of the protons, and then the other leftover proton is just released, right, as an H+. Right? So in doing so, this nicotinamide adenine dinucleotide, or this NAD+, is serving as a convenient place for some big enzyme working on a substrate, taking a pair of electrons off that substrate to just dump those electrons onto this NAD, turning it into NADH, and the enzyme is still free to keep working, right? And then this NADH would float off. So these coenzymes are super important in order for certain enzymes to even work, right? As a result, many of these coenzymes are what we commonly everyday call vitamins, right? You might have wondered, well, you know, vitamins are important. You always need vitamins. You might take vitamin supplements, but what are vitamins? What do they really do, right? Many vitamins are coenzymes. Here's an example here. Here's vitamin B3 or niacin, right? So here's the structure of the vitamin B3. So that would be the part that would be like up here on some electron acceptor molecule, right? So in other words, vitamin B3 is important because in certain enzyme pathways, certain enzymes would need this as the electron acceptor. All right, so let's put this together into a everyday kind of example to help you understand many of these complex concepts, right? So alcohol, we were talking about the oxidation of alcohol. So here you have ethanol, for example, in beer or wine, right? So ethanol is toxic to our bodies. Our bodies have to neutralize the ethanol by breaking it down. In other words, by oxidizing it, right, into harmless little side products. So a whole series of enzymes, a whole series of proteins are involved in a step-by-step fashion. The first enzyme is this ADH, or alcohol dehydrogenase. So here it is, right? Here's the shape of it here. But look at this cartoon. This alcohol dehydrogenase is the right shape to bind the ethanol and one of these NAD+ coenzymes at the same time. So as a result, the ADH, the big purple protein, takes electrons off of the ethanol, thereby oxidizing the ethanol and instantaneously transfers those electrons over to the NADH coenzyme carrier, right? Thereby releasing both of the products. And one of the products is now acetaldehyde, right? And importantly, the ADH protein is not stuck hanging on to electrons. So it can go to work again on another ethanol with another NAD+. So from that acetaldehyde, right, another protein, aldehyde dehydrogenase, right, is the right shape to bind the acetaldehyde and another coenzyme NAD+, and it goes ahead and it converts the acetaldehyde into acetate. And then that acetate, this goes on and on and on. Another series of enzymes eventually break it down to carbon dioxide and water, right, which we just release as waste, right? So the ADH converts the ethanol to acetaldehyde, which ironically is actually more toxic than the original ethanol, right? So your body has to very quickly break down the acetaldehyde to deal with it. Right? So fortunately, the ALDH in orange here quickly converts the acetaldehyde to acetate and then eventually down to water. So here's kind of an interesting story. You know, myself being Chinese, you know, my whole life, or I should say ever since I was of drinking age, right? When I drink beer or wine, I turn a very nice rosy red hue, right? And I remember in college, we used to drink and my friends would be like, oh, you know, look at Howie, he can't handle, you know, his beer, right? Because I'd get all messed up looking. But I'd tell them, hey, I actually feel fine, except I feel like, you know, yeah, my eyes are a little runny. You know, I have a flush, right? This is the whole Asian flush thing. But I actually would feel really fine other than that, right? So what's going on there? So it turns out that about 80% of East Asians have a variation in the coding sequence of the ADH gene that codes for this alcohol dehydrogenase, right? As a result, the shape of that protein actually works better at converting the ethanol, right? So you convert more ethanol over to acetaldehyde a lot faster, right? And you say, well, but that doesn't make sense because I thought, you know, the whole point was you don't somehow metabolize the alcohol faster. But this is the key here. Around 50% of those same individuals also have a variation in the ALDH gene that codes for the aldehyde dehydrogenase protein. And that slows down the conversion of the more toxic acetaldehyde into acetate. So as a result, the symptoms you get, right, are that you actually have allergic reactions to the acetaldehyde. And that is your skin getting flushed, you start having a runny nose, you know, eyes get runny, those types of things. But interestingly, you don't feel as much buzz because you've actually converted the alcohol much faster. So that's why I often told my friends, hey, you know, I could drink a lot more than my friends in college, even though I just looked like a mess, right? But it took me a lot more in order to actually feel the effects of the alcohol. But unfortunately, I had to suffer the effects of the acetaldehyde, right? Okay. So that's it for today's lecture. But hopefully that example helps put together some of these concepts, right? How our bodies are currently breaking down things through oxidation, things like ethanol, right, that our body needs to get rid of. It breaks it down through a series of different enzymes that have evolved, coded for by these different genes that have evolved, right, to make these proteins that then go to work. And in a stepwise fashion, each one has a very specific substrate that it works on and does some sort of catalytic function to convert that substrate into some product. And then it's up to some other enzyme now that recognizes that product to then do its job and convert that into another product. And in the case of ethanol, going through a whole chain of enzymes, eventually neutralizing the ethanol into carbon dioxide and water, right? And each one of these enzymes along the way needs energy in the form of ATP to be able to go ahead and drive these different reactions. And importantly, like the last thing I want to mention, because I know I'm over time here, is think about this. Each one of these enzymes as a protein is coded for by its own gene in your genome, right? And different variations in the genetic sequences that are associated with things like different racial ethnic backgrounds, for example, right, can result in different, slightly different functioning versions of these proteins, right, that may alone not have a big effect, but when taken together in the context of the particular biochemical cellular pathway that they all contribute to, can actually have big effects, right? All right, let's go ahead and stop there today. Let's go to the chat. Okay. All right, yeah, I know that my whole story about the whole drinking thing, that always gets some reactions here. Okay, so it looks like there are no other questions related to the material here. So hopefully, my honest hope is that you guys find this really interesting, right? Because what we're trying to do is actually relate things like your genes to eventually different proteins that actually carry out the functions inside of your cells, right? And these are just some good examples of important types of functions that certain enzymes have, right? But it's actually wonderful to think, right? This is just one specific example of a pathway that exists in your cell, right? Think about all the multitude of other complicated pathways that also involve different proteins, each playing their role, right? And how this contributes to your cell constantly being a mix of different pathways building things up, while at the same time, different pathways breaking things down, right? And this all requires the input of energy at the top in order to get all these things to happen, right? And that energy comes from glucose. And we get that glucose from the food we eat, right? And the food we eat came from other animals, other plants that originally put together that glucose using the energy from the sun, right? All right, so I hope you guys have a great weekend, and I'll look forward to seeing you on Monday, all right? Okay, bye-bye.